What we're going to do right now, I think I'm going to change direction, move away from drugs or surgery and move more into the area of nutrition, evolution, nutrigenetics, nutrigenomics. Let's see now, this thing moved while I was not. Let's see. I need to go backwards. It moved on its own.
This is backwards.
This is the next one. This is the current one.
No.
This is the next one. This is the current one. So when you choose this,
Right.
this is what they see, and this is
but
what's coming.
I don't want to use, excuse me?
Try
I
this.
don't want to use this.
Oh, yeah.
So forward is which one?
This one.
The under and the top.
Okay.
All right, very good.
There's only the pointer.
Yeah, thank you very much. So it has been for many, many years, almost 100 years, that people had examined the influence of fat in cancer development, not only breast cancer, but other forms of endocrine cancers. It was not, though, until about 20 years ago that people very seriously began to look at the various types of fatty acids, and particularly at the essential fatty acids, the omega-6 and omega-3 fatty acids, and their role as well as the ratio of omega-6 fatty acids to omega-3 in growth and development and their role in chronic diseases. So it became very obvious that in terms of the evolutionary aspects of diet, human beings evolved on a diet that was much lower in saturated fat than the current diets, and it had equal amounts of omega-6 and omega-3 fatty acids, omega-6 in red, omega-3 in blue, and that major changes began to take place about 200 years ago, and then the last, I would say, 70 years, there's been practically deletion of omega-3 fatty acids from the diets of people worldwide, and particularly in Europe, the United States, South America, and China. At the same time, there has been an increase in the amount of trans fats, which now we're trying to get rid of, and definitely lower amounts of vitamin C and vitamin E. With all these changes, there have never been any changes in our genes. So our genes were programmed to respond to an entirely different diet, particularly in terms of the essential fatty acids and antioxidants, than the diet that they're being faced with at this point. So what are some of the consequences of this change? Many studies have been carried out in indigenous populations, so we know, I honestly don't know what to do. It keeps on moving on its own. All right, now I'm where I want to be. So what we have when we're looking at the arachidonic acid is the omega-6 fatty acid that is the parent fatty acid for the development of prostaglandins, leukotrienes, and EPA is the omega-3 fatty acid that is responsible for the production of also thromboxanes and leukotrienes. And when you look at the level of the platelet or the endothelium or the neutrophil, the eicosanoid metabolism from arachidonic acid actually, in terms of the thromboxanes leads to vasoconstriction and pro-aggregatory properties, where those of EPA and DHA are exactly the opposite. And the same thing appears to be at the level of the neutrophil, where at the endothelium, their functions are the same. But what I wanted to go over, and I'm not sure what happened to that slide, is that a series of studies have been carried out in terms of the ratio and amounts of linoleic, which is the omega-6 fatty acid, versus ALA, alpha-linolenic acid. During the Paleolithic period, the ratio of LA to ALA was about 1 or just less than 1. This is because under normal conditions and during evolution, the amount of alpha-linolenic acid in green leafy vegetables was always much higher than that of linolenic. And most of the seeds available under natural conditions were much higher in alpha-linolenic than linolenic. In Western diets, because of the changes that took place in terms of animal feeds, being fed grains, which are high in omega-6 fatty acids, and the production of oils such as corn oil or soybean oil, sunflower oil. These are extremely high in omega-6 fatty acids. For example, the ratio in sunflower oil is 77% omega-6. So the ratio is about 77 to 1 practically, instead of being balanced or 1 to 1. So all of a sudden, we present our cells and our physiology with extremely high amounts of omega-6 fatty acids, while at the same time, the omega-3s are extremely low. So Western diets today then are characterized by extremely high amounts of omega-6 and a high omega-6 to omega-3 ratio, both at the level of 18 carbon atoms as well as the longer chain fatty acids. So the total, this is from in the US, ratio in terms of the current food supply is 16 to 1. Frankly, there are a lot of data that they show this ratio is much higher. I know in Europe is about the same as in the United States. So why are we actually concerned about all this? Let me go over the metabolism. So here we have the omega-6 fatty acids, which is the linoleic acid, the omega-3 fatty acids, which is linolenic acid. These are the parent fatty acids. Normally, you have more omega-3 than omega-6. This pathway is stronger than the omega-6. Both pathways, they use the same enzymes, both in terms of desaturases and elongases. And there are genetic variants at the level of delta-6 desaturase and the delta-5 desaturase, which have been very well worked out. And there are, during the process, then if you have, in today's diet, this pathway unfortunately overwhelms the omega-3 pathway. So that have a much higher production of arachidonic acid, which, as I showed you earlier, is the parent fatty acid for the development of the series of thromboxanes and the 2 series of prostaglandins via the COX enzyme, which aspirin blocks and statin blocks, and arachidonic acid through the 5-lipoxygenase produces leukotrienes, which are extremely pro-inflammatory. So out of the arachidonic acid, you have a very pro-thrombotic and pro-inflammatory pathway. And this, which is not so much pro-inflammatory, is actually exists in very small amounts and does not work all the time. Whereas in the omega-3 fatty acids, again, the eicosanoic acid through the COX-2 produces much, I would say, less thrombotic series and less inflammatory series, but both enzymes, COX-2 in this pathway, as well as COX-2 in the omega-6 pathway, they can be polymorphic. So depending on the polymorphism, you may have different products. And it's important whenever you want to determine the fatty acid composition of the dietary intake, that actually you measure the metabolites, or you measure the ALA, arachidonic acid, EPA, and DHA directly on the red cell membrane phospholipids, because dietary history, it only gives you a small indication of what people eat, since there is endogenous production of EPA and DHA based on the genetic variants of the enzymes of the delta-6 desaturases that may account as much as 30%. So I pay no attention to the dietary history. If you are going to do a study and you want to evaluate the effect of a drug or physical activity, or you measure behavior, it's important to have very good information. What is the diet prior to, as well as during the intervention? Dietary intakes, when it comes to the determination of fatty acids, are inadequate. You need to measure the red cell membrane phospholipids, which gives you an idea what the person has been eating for about 120 days. Or some people go ahead and do biopsies of the adipose tissue, which gives you information what the situation has been in terms of 3 or 4 years. The reason I went through the pathways and pointed out to the different metabolites is because they have not only opposing properties, the omega-6 being pro-inflammatory and pro-thrombotic, but at the same time, a high amount of linoleic acid in the diet blocks the incorporation of EPA from fish oil or eating fish. That's another evidence that the dietary history will not tell you what's going on in the body. So whatever you do in cancer intervention studies or cardiovascular studies, it is very important to understand and know the composition of the diet prior to the study and subsequently, because all chronic diseases are pro-inflammatory. They have pro-inflammatory components. The basis is inflammation. The omega-6 fatty acids that are in high amounts in our diet, they are the most inflammatory agents that we ingest. And I think this is a very important point, particularly when we know that cancer is an inflammatory disease. Let me see now. So I was particularly interested. I knew that in terms of their physiology, the omega-6 and omega-3 fatty acids are very different. The question is, how do they influence gene expression? And here I pulled data from a number of studies. And what we have here is the effects on the platelet, which I went over with you previously. And so we took linoleic acid, omega-6, alpha-linolenic acid, which is omega-3, arachidonic acid, omega-6, EPA and DHA, omega-3. And we studied the effects of PUFA on several genes that encode enzymes, proteins involved in inflammation, adhesion molecules, cell growth, early gene expression, beta oxidation, and growth factors. Because the question was, are all their effects always opposing, or there are situations where their effects are similar? It might even be additive. So you can see that in terms of the effect of fatty acid on cell growth and early gene expression, including genes involved in early cancer development, the arachidonic acid increases the gene expression, increases the mRNA, where EPA and DHA suppresses it. in terms of the adhesion molecules, which are definitely increased in cardiovascular disease, the arachidonic acid has a mild effect, but the DHA has a very prominent effect and really suppresses adhesion of molecules, and that's one of the factors why DHA, along with EPA, is important in the management of cardiovascular disease. Now, inflammation, interleukin-1 beta is one of the most important cytokines. Arachidonic acid increases the production of interleukin-1 beta, where EPA and DHA suppresses. But in cases where we're dealing with beta oxidation, the mRNA, the expression of the gene, the mRNA increases in both omega-6 and omega-3 fatty acids, all of them. But again, the DHA appears to be more potent. When it comes to growth factors that are important both in heart disease and cancer, their effects are opposite. So the majority of the physiologic effects of essential fatty acids between omega-6 and omega-3 are opposite, but not always. So what are some of the possible roles of omega-3s through which we can protect many of the hallmarks of cancer? And there have been tissue cultures and animal experiments where the omega-3 PUFAs, they have inhibit proliferation of cells, where the omega-6 fatty acids, by the way, increase proliferation. They decrease tumor invasion and metastasis, many animal studies like that. They reduce the oxidative stress, and the replicative ability is blocked and leads to genome stability. And it may actually improve mechanisms through which DNA is being repaired. And they normalize obesity-induced metabolic profile, and there are many, many studies on that. In fact, I noticed earlier today, people mentioned the Women's Health Initiative, which is one of the best group of studies at the NIH, and they specifically looked at the relationship of the omega-6 and omega-3 fatty acids relative to obesity. And these are very good studies because they have very accurate data, and they have red cell membrane phospholipid levels on all the omega-6 and all the omega-3, and they were able to show for the first time that a higher ratio of omega-6 to omega-3 or a higher omega-6 leads and increases the risk of obesity in healthy women 10 years later. And so this is one, I think, of the most important studies because it is accurate and it is done very well, and the women who are followed at various intervals, not at the beginning of the study and 10 years later. And the increase in risk of obesity is significant, which is exactly what had been shown over the years in many animal studies, but not in human beings because the dietary histories were not adequate, and they were using data in obesity where they were self-reported rather than actually measured. So if you do the study correctly, if you have biomarkers, if you had measurements on the red cell membrane phospholipids, if you measure the weight and the height, or the BMI of the patient, a high omega-6 to omega-3 ratio, as it is today in all Western cultures, increases the risk of obesity in women. And obesity, of course, increases the risk of cancer. Additional mechanisms show very clearly that the omega-3 fatty acids, they suppress the biosynthesis of pro-inflammatory molecules. They modify transcription activity and gene expression, I showed you that. They influence signal transduction, and there's an alteration of hormone-stimulated cell growth, suppression of production of free radicals, and influence insulin sensitivity and membrane fluidity. And this may be one of the most important factors in terms of their anti-inflammatory effects and their anti-cancer effects. As I said, DHA appears to be more potent than the other omega-3 fatty acids, and there is very good evidence that DHA exhibits cytotoxic effects on cancer cells in vitro and in vivo through inhibition of proliferation and migration of tumor cells, induction of apoptosis, inhibition of inflammation and angiogenesis. This has been known for years. And induction of immune responses. So when you think of immunoprevention, immunotherapy, DHA is one of the most important factors to consider and take into consideration. So there have been a number of studies that I don't have time to cover in terms of the influence on gene expression, transcriptome factor activity, and signal transductions, and in essence, a high omega-3 to omega-6 ratio, which is really what we want to have. We want to go beyond the balance. For normal health, you need to have equal amounts, but when you treat diseases such as heart disease, cancer, arthritis, ulcerative colitis, you need to think of much higher amounts of omega-3. And so they increase apoptosis, they decrease cell proliferation by blocking the various factors shown here. And in terms of cell apoptosis, they really decrease P1K, PACT, nuclear factor kappa B, while they're very important in terms of increasing PPAR gamma, and they actually decreasing the ratio BACL2 to BAX. This has been shown repeatedly by various groups, and it's something that is now very well accepted. So the question is, what is the role of omega-3 fatty acids in terms of cancer prevention? And tamoxifen has been certainly used for many years, and you had the excellent presentation earlier today. So this is a study that is done in animals, where they wanted to see what would be the effect if, in addition to tamoxifen, they added equal amounts of fish oil and corn oil versus tamoxifen with high corn oil, which is what the studies on tamoxifen today have been done on a population with high amounts of omega-6. So this is the animal model where they took groups of female spray rats. They were injected intraperitoneally with 1-methyl-1-nitrosourea at 50 days of age and assigned to either a controlled diet that was 20% corn oil, very similar to Western diet, or a fish oil-rich diet having equal amount of fish oil and corn oil in the presence and absence of tamoxifen in the diet, 0.6 part per million. The rats were sacrificed at weeks 4 before any palpable tumors, and again at 8 and 12, where 90% of control rats already had palpable tumors. The results demonstrate a major effect of tamoxifen in inhibiting the development of early preneoplastic lesions, which we know from the human studies. Fish oil, while having a marginal protective effect of its own, enhanced the anti-tumor action of TAM on all histologic parameters of carcinogenesis, although the effects of the combination were not statistically different from those of TAM alone. The combination of fish oil and TAM was the only intervention that induced regression of established preneoplastic lesions. I think this is the most important aspect of the studies. They also found that the contrast tissue biomarkers, they also found that in contrast to plasma, only target tissue omega-3 fatty acid levels correlated with select tissue biomarkers of carcinogenesis, whose expression was altered in a manner predictive of a protective effect. So these results demonstrate the potentially superior chemopreventive efficacy of TAM and omega-3s have important translational implications. The data also emphasize the importance of local factors affecting target tissue levels. And this is very important, because if you do studies and you measure the amounts of metabolites in the blood, that does not always tell you what is going on at the tissue level. This experiment was very easy to show that what made a difference was actually the amount of omega-3 fatty acids at the tissue level. And I can tell you there are studies that have been done in Canada in patients with prostate cancer, where they have been able to follow patients with prostate cancer that did not have to be operated, but they followed to see the changes that occur. And the amount of omega-3 fatty acids in human studies at the level of the tissue of the prostate was very significant in protecting or delaying the development of the prostatic cancer. So this study is a very important study, and it definitely actually collaborates the effects that one has seen from tamoxifen over the years. And I think a combination of tamoxifen and omega-3 fatty acids is something that needs to be considered in terms of chemoprevention. So there is another mechanism that I want to emphasize. You know, the omega-6 fatty acids, we said, are pro-inflammatory. And they produce prostaglandin E2, which is prothrombotic, and LTB4, the leukotriene LB4, which is pro-inflammatory. And they both lead to chronic inflammation, which has been linked to mutagenesis, mitogenesis, angiogenesis, anti-apoptosis, and metastatic factors associated with cancer initiation and progression. The omega-3 fatty acids alter estrogen metabolism by suppressing the production of PGE2 from arachidonic acid. So if you have a diet that is high in omega-6 fatty acids, and so it's high in arachidonic acid, it produces high amounts of PGE2, which stimulates the activity of aromatase P450, giving inhibitors to aromatase, and converts 19-carbon steroids to estrogens. You're not going to get the effect you would have gotten if the background diet was balanced in the omega-6 and omega-3 essential fatty acids. On the one hand, you give aromatase inhibitors, and on the other, we have a diet that is high in omega-6, which increases the production of what the inhibitor is trying to suppress. You can see then, if you don't know the amounts of omega-6 and omega-3 in the diet, when you do studies in different populations, you're not going to get the same effects. Never mind the genetics at this point. Just think in terms of the diet and the interaction of the drugs with that particular diet. So the question is, what happens? In the US, people take a lot of supplements, and they wanted to be sure that they can go back and look. And these are not necessarily the best possible studies, but that's the only thing they could do. And these are in a group of women who were taking part in the vital cohort. They were actually analyzed specifically for two factors. One was vitamin D and the other omega-3 supplements. And they found that on the average, 10 years later, there was a suggestion that the omega-3 fatty acids were inversely associated with cancer of the breast. In other words, the higher the amount of the omega-3 supplements that they were taken, the lower the risk of breast cancer. But I don't think it's really one of the best studies, but this is what most of the studies funded by the National Cancer Institute are like. So there have been a number of studies from England and the US done by epidemiologists and schools of public health that they have never shown any relationship between omega-3 fatty acids, EPA and DHA, and cancer. Of course, just think what sort of a diet we have in these countries, in UK and US. These people, of course, they did not actually measure the amount of EPA and DHA in the diet. However, a very important study took place in Singapore. These were the results of a progressive study of women in Singapore where fish intake is much higher than that of the US, showed an inverse association between dietary omega-3 PUFA from marine sources and breast cancer risk. The only cohort studies in which individual associations of EPA and DHA intake from the diet with risk of breast cancer have been examined are the Nurses' Health Study, which is at the Harvard School of Public Health, and the Netherlands cohort. In these two studies, no association was found in either study. But if you go back and see how the studies are done, I wouldn't expect to find any association. These are dietary data, self-measurements, and the nurses are called in or are seen again every 10 years. So in contrast, in another meta-analysis in which blood biomarkers of fatty acids in association with breast cancer risk were analyzed, they found inverse associations with total omega-3 PUFAs and so on. For all but EPA, the association persisted when the analysis was restricted to postmenopausal women. So again, unless the studies are done properly, you're going to find conflicting results. And we need to really maybe do a little bit more accurate studies by looking at genetic variants and blood biomarkers, and try to move away from epidemiologic studies done in different countries based on dietary history. So we know, for example, that in breast cancer, lipoxygenase 5, and I showed you that enzyme is involved in the metabolism of omega-6 and omega-3, and lipoxygenase 5A binding protein are overexpressed in tumor tissue and have been related to cancer prognosis and survival. So both of these mutations and both of these enzymes are part of the omega-6 and the omega-3 pathway, but they are in high amounts because of the high amounts of omega-6. So the question is, disruption of signaling pathway in lipoxygenase 5 mediates growth arrest and apoptosis in breast cancer cells and inhibits breast cancer metastasis. So the question was, with this information, whether one could look at a group of women and trying to associate or look with any relationship between the genetic variant of the lipoxygenase or the lipoxygenase binding protein and the risk of cancer. So he conducted a population-based case control study among 3 different groups in the Bay Area in California of African Americans, Spanish, and white Americans, and looked specifically for a relationship between dietary linoleic acid and breast cancer. So, however, he did not find any relationship between the presence of the mutation and the evidence of cancer. However, when they looked specifically at the genetic variant and its relationship to the amount of the linoleic acid that was being consumed, they found out that those women who had the genotype of AA rather than AG or GZ and consumed more than 17 grams per day, they actually did not have cancer, but those who had the genotype AA, it was associated with a higher breast cancer risk. Now, this is a very important study because it shows having the mutation, it may have no effect. So the question is, what are the factors that interact with that mutation that may actually produce the effect? Because we know that these mutations occurred in enzymes that are involved in the pathway of the omega-6 and omega-3, they look specifically then for the genotype in the mutation and related to the amount of omega-6 fatty acids. Now, 17.4 grams of omega-6 fatty acids is about 20% of energy, which is very high. The American Heart Association keeps on increasing the amount of polyunsaturated linoleic acid in the diet and recommended to go up to 10%. I think this is a mistake because depending on the population, an increase in linoleic acid is going to increase the risk of cancer in these women. So the findings then support a role for omega-6 PUFA in breast carcinogenesis and suggest that epidemiologic studies on dietary fat and breast cancer should take into account genetic predisposition related to omega-6 PUFA metabolism. So I'm going to move into conclusions. And results from in vivo and in vitro human studies suggest that omega-6 fatty acids, they accelerate tumorigenesis, whereas omega-3 fatty acids have anti-cancer effects. Alpha-linolenic acid, EPA, and DHA can differentially inhibit tumor development by changing the cell membrane fatty acid composition. They suppress arachidonic acid-derived eicosanoid biosynthesis and influence signal transduction pathways to inhibit cell proliferation and induce apoptosis. Therefore, using omega-3 PUFA as a nutritional intervention in the treatment of cancer could enhance conventional therapeutics or potentially lower the dose. In order to provide definitive recommendations, additional human studies are required. Long-term studies tracking fish or omega-3 PUFA intake are needed to demonstrate a role for omega-3 fatty acids in prevention. Also, additional clinical trials are needed to evaluate the effect of omega-3 PUFA on cancer outcomes. Nevertheless, evidence does not indicate harm, and all forms of omega-3 PUFA may be included in a healthy diet. Omega-3 fatty acids have important anti-cancer effects and warrant further study as preventive, decrease the risk of cancer development, and therapeutic along with chemotherapy agents. Thank you very much.